it would have also been nice to come out and see the campus and visit Newfoundland. I haven't been there before, but I've heard amazing things. And yeah, so just generally a big thanks to everybody for coming out. Okay, so as Cole mentioned, I'm going to talk a bit today about mechanisms and costs of change. And so over the course of today's talk, I really want to share with you some of the tools that I'm using to dissect out phenotypes that allow animals to deal with one of the most challenging environmental factors that they face. And again, again, that's change. So realistically, all environments, sorry, can I just ask really quickly here? Can you guys see the slide properly, or is the Zoom thing like over top of it?
No, it's looking good. Yeah.
Okay,
Looks good on
cool.
my end.
All right. Yeah, so realistically, all environments are experiencing some degree of change. But we're really at a point in global history where we are seeing increasingly rapid and complex changes in habitats across the globe. So things like warming oceans, coral bleaching, deforestation, and increasing rates and levels of anthropogenic pollution. And these provide some examples. And there are pretty alarming examples from a conservation standpoint, but it can also be a bit interesting from a mechanistic standpoint, because it really lets us kind of dig down and really understand how animals contend or cope with these novel challenges. So change can be really difficult because it challenges how animals can maintain steady states. So I have a performance curve here. We have an environmental gradient on the x-axis here, and we see that over an environmental gradient, performance can be described by a quadratic curve. So here, environment can represent anything. It could represent something like abiotic environments like temperature, salinity, oxygen concentrations, or biotic factors like food availability, disease, density. On the y-axis here, we have performance. Again, this can represent a lot of things that span levels of biological organization. So you could have something all the way down to rates of transcription on kind of a molecular level, all the way up to tissue level responses. So oxygen consumption of specific tissues, or even whole animal performance responses. So things like whole animal metabolic rate, locomotive capacity. Yeah, so really in terms of performance curves, we can look across environmental gradients and across different types of performance. So what we generally see is that all animals have evolved to optimize their performance to their local environmental or their local conditions. But as animals move outside these local optima in space or time, we tend to see a reduction in performance. So I like to use temperature examples because a lot of my background's in thermal physiology. So essentially, if we think about this environmental gradient as a thermal gradient, as animals move into colder environments or the environment around them becomes colder, what we see is a slowing down of biological rate processes. So for example, the metabolic pathways that produce ATP rely on a series of reactions catalyzed by enzymes. So if those enzyme reaction rates slow down with temperature, what we would expect to see is a decrease or a limitation in the amount of energy animals can produce in cold environments. That being said, in many animals, performance curves can be plastic. So animals can remodel their physiology, and this allows them to overcome these changes in their environment. And this, as you can see in the figure, is represented by a shift in the thermal performance curve. So we see that it's shifted leftward here to optimize performance to this new environmental temperature. So essentially, we can think about plasticity as a flexibility, where physiology can be remodeled in response to the environment. And often these changes are beneficial to performance, in which case they are termed adaptive plasticity. So for the remainder of today's talk, when I mention plasticity generally, I'm specifically referring to adaptive plasticity. And again, if we go back to our thermal biology example, even though, say, an individual in this, so originally this is the warm acclimated level of performance. So even though we have an individual that's now been cold acclimated to this colder environment, the acclimation response can come through mechanisms, so say something like mitochondrial biogenesis. So even though specific metabolic pathways are operating at slower rates, animals can expand the number of pathways operating in parallel by producing more mitochondria, more metabolic enzymes, and thereby maintain metabolic flux despite these thermodynamically limiting environments. So that's an example of an acclimation response or an adaptively plastic response. So there's generally two types of compensatory plasticity or adaptive plasticity. We have developmental plasticity. This is where environmental cues during development determine subsequent phenotypes. So this process has generally been described as irreversible. In the last decade or so, I think there's been a bit of pushback against that some developmentally plastic processes are technically reversible, but in the general sense, these like major kind of classical archetypes that we see. So in the case of these Daphnia here, where we get kind of the standard phenotype in the absence of predatory cues, but this adult phenotype with the spiny helmet in the presence of predatory cues, what we tend to see is that's an irreversible change. So this is permanent. So if upon maturity, we took this spiny helmet phenotype and returned it to waters with no predatory cues, we wouldn't see a reversal of this phenotype. So I'll mention this now, but it comes in like a little bit later, is that in that case, we'd see something like a mismatch cost, where this individual has dedicated energy and resources to creating the spiny helmet, which may represent a cost if there aren't actual predators in their environment, which it'll ultimately defend against. The other kind of adaptive plasticity is reversible acclimation. And this is exactly what it sounds like. It's a reversible remodeling of physiology in response to changing conditions. So in the case of the rainbow trout that I'm showing here, we see remodeling of the heart to increase contractile function in cold conditions. So we actually see hypertrophy of the heart tissue in cold acclimated rainbow trout, and that's going to counteract the limitations of cold environments on muscle contraction, cardiac muscle contraction, so that these cold acclimated fish can maintain cardiac scope. The thing here is that if we return these cold acclimated fish to a warm environment, we see a reversible remodeling where the heart then returns to its original size. So again, the main difference between these two things is that developmental plasticity is generally thought to be irreversible, though there's some debate surrounding that. And it prepares animals by matching mature phenotypes with either current or predicted future environments, whereas reversible acclimation is more of like this dynamic processing, where there may be a bit of a lag time, but animals can kind of catch up their physiological phenotypes to the direction that the environment is shifting in. So there's often interspecific genetic variation for plasticity. And this means that selection can act to shape the amount of plasticity that different populations exhibit. So in variable environments, we'd expect to see a high degree of plasticity because we'd expect plasticity to be buffering against these dynamic changes. In more stable environments, however, it appears that plasticity may come at a cost. And this is because a number of studies have demonstrated a phenomenon called genetic assimilation. So this is essentially a pattern where populations in more stable environments show less plasticity. And again, I'll mention that it's been difficult to tease apart whether or not this truly reflects cost of plasticity as opposed to genetic drift. So even though there's been a lot of theoretical work surrounding this, we don't have a ton of empirical evidence regarding the cost of plasticity. So we don't especially know if plasticity has costs or what those costs entail. And this really makes it difficult to predict whether or not populations will persist when they're faced with the increasingly rapid and complex environments that we as a species are creating. So one of my main interests deals with identifying what the costs of plasticity are. And this is where I find that the integrative approach that I'm taking can really be quite powerful. So here we have the basic pipeline from genotype to phenotype to whole animal performance, and then scaling all the way up to fitness. And in simple terms, what plasticity really is a gene-environment interaction, whereby large-scale gene expression programs are regulated to maintain performance. And I really work from the bottom up, where I study the most basic functional levels of physiology in order to start identifying proximate mechanisms that regulate these plastic phenotypes. And kind of for the purpose of today's talk, I'm going to refer to these molecular mechanisms or these proximate mechanisms as levers. So in other words, when animals are faced with stress, there's a finite number of molecular levers that they can pull to coordinate these compensatory phenotypic responses. And by identifying what these levers are, we can really start to make predictions about associated fitness costs. So things like maintenance costs. So what is the cost of maintaining the levers themselves, or ecological costs? So what happens when a lever compensates or allows an animal to overcome one stressor that makes it more vulnerable to a second or interacting stressor? So these are things that I'll talk to in today's presentation. It's kind of split up into two parts. The first part dealing with mechanisms, where I'm going to talk about some of my PhD work looking at regulatory pathways for thermal acclimation in ectotherms. So again, what are these molecular levers? And then shifting over into a discussion about costs. So first looking at ecological costs of stressor interactions. So what happens when these levers are pulled in different directions? And what are the costs of adaptive plasticity, but particularly when that plasticity is not being used? So what are the costs when these levers start to rust? So during my PhD, I was looking for upstream regulatory pathways for thermal plasticity in ectotherms. So I focused on zebrafish as an ectotherm model, and I focused specifically on thermal plasticity. But really I'm looking in the bigger picture, trying to identify what are the regulatory mechanisms that integrate environmental cues with molecular and cellular responses, so for plasticity in general. And I kind of focused right in on thyroid hormones after reading the literature, because we already know that thyroid hormones promote thermogenesis, or metabolic heat production, in birds and mammals, specifically in response to cold temperatures. And we also know that they act on the same levels of physiology that characterize the thermal acclimation responses in ectotherms, so specifically looking at zebrafish. The big question here was whether or not thyroid hormones coordinated these pathways supporting responses, specifically in changing thermal environments. So this is the first kind of experiment that I did as a PhD student with Frank Seebacher at the University of Sydney in Australia. And I was first looking at just a general thermal acclimation response in an important whole animal trait, and that's swimming performance. So on the x-axis here, we have acute test temperature. This is the temperature that these fish were swam at. On the y-axis, we have critical swim speed, so UCRIT. This is essentially like a beep test for fish. So we have the fish in its flume, it's swimming against a current, and every 10 minutes we'll turn up the current a proportion of the fish's body length. And we'll do that till the fish becomes physiologically exhausted and can no longer maintain its position in the water column. In the blue, we have cold acclimated control fish. So these fish were acclimated to 18 degrees Celsius for 3 weeks. And we have warm acclimated control fish, which were held at 28 degrees Celsius also for 3 weeks. So the first thing I want to point out here is if we compare the warm acclimated fish swam at 28 degrees Celsius to the warm acclimated fish swam at 18 degrees Celsius, we can see that Q10 effect. We see the decrease in performance that's an acute effect of temperature. Now, when we look at the cold acclimated fish swam at 18 degrees Celsius, we can see that they swim significantly better than the warm acclimated fish. I shouldn't say better, I should say they swim significantly faster. And when we compare the cold acclimated fish swam at 18 degrees to the warm acclimated fish swam at 28 degrees, what we see is this almost perfect acclimation response, where the cold acclimated fish have almost 100% compensated for the limiting effects of their colder environment. So to specifically test the effects of thyroid hormone as a regulatory pathway in this response, I also introduced pharmacological hypothyroidism. So essentially I drugged the fish to inhibit thyroid hormone production and also metabolism in peripheral tissues. So we see immediately that in the cold acclimated fish swam at 18 and 28 degrees, we see this huge reduction in swimming performance, whereas we see relatively no effect in the warm acclimated fish with hypothyroidism. So again, hypothyroidism in cold acclimated fish in dark blue, in warm acclimated fish in dark red. So we see that cold acclimated fish are much more sensitive to hypothyroidism treatment, and warm acclimated fish are relatively insensitive. And what this is really suggesting is that thyroid hormone is specifically playing a role in this cold acclimation phenotype that we were seeing. And I also had another treatment where I took the cold acclimated hypothyroid fish, and I also treated them, supplementing them with thyroid hormones. So this was kind of a control to make sure that the hypothyroid effects we were seeing were a direct effect of thyroid hormone levels and not some off-target effect of the 2 pharmacological agents that I was using. So when we treat the cold acclimated hypothyroid fish with thyroid hormones throughout the acclimation process, what we see is a complete recovery of swimming capacity. So again, indicating that thyroid hormone is regulating this cold acclimation response. So for the rest of my PhD, I was really interested in potential mechanisms underlying the effects we were seeing. So what exactly is thyroid hormone doing to help promote and maintain swimming performance in these otherwise limiting environments? So for this, I looked across levels of physiology, looking at aerobic metabolism, skeletal muscle function, and cardiovascular function. So I've put up all the different measures I took across these levels of physiology, kind of spanning upwards in terms of levels of biological organization, where I looked at things all the way from isoform expression levels, so like transcriptional rates of different enzyme isoforms, all the way up to things like whole animal metabolic rate, of course, critical swim speeds, which I just showed you, and kind of regulation through the central nervous system. So the effects of thyroid hormone on the central nervous system's kind of capacity to help shift to these different phenotypes. So I've highlighted the ones in green just as a time limitation that I'll talk to you about then, but I've also thrown up citations here. So if any of these things interest you, by all means go and check it out in the actual papers. So looking first at aerobic metabolism, we see that cold acclimated fish, and this is reminiscent of the swimming stuff that I just showed you, have significantly higher aerobic scopes than warm acclimated fish. So this is in fish measured at 18 degrees Celsius. We have aerobic scope on the y-axis. So aerobic scope is essentially that difference between resting metabolic rate and maximal metabolic rate. And you can almost think of aerobic scope as being this energy reserve that fuels all fitness related performance. So it essentially is telling you what your energy budget is for things like swimming speed to escape predators, to chase prey, for social behaviors, mating, reproduction. All of these things take energy out of this aerobic scope energy budget. So again, we see very reminiscent of the swimming performance is that when we make these cold acclimated fish hypothyroid, we see a significant reduction in aerobic scope, whereas we see relatively no effect in warm acclimated fish. And then if we look kind of down a few levels of biological organization, or look upstream, I should say, at mRNA expression, we see a very similar pattern. So just to orient you on this graph here, on the x-axis we have genes encoding different subunits or different proteins involved in metabolic regulation. So PGC1 alpha here is known as the master regulator of metabolism in vertebrates, whereas these other genes here all encode subunits of enzymes involved in electron transport and ATP synthesis in the mitochondria. On the y-axis here, we have the relative mRNA level. So again, we can see cold acclimated fish have increased the expression, or at least the transcription of these genes. But when we make them hypothyroid, again, we see a decrease in expression or transcription levels of cold acclimated individuals, again, with relatively no effect in warm acclimated fish. So this is really indicating that thyroid hormone is acting transcriptionally to upregulate metabolism, specifically, presumably to offset deleterious effects of cold environments on kind of ATP synthesis pathways. So looking from a skeletal muscle angle, we also see that cold acclimated individuals are capable of higher tail beats frequencies. So on the x-axis here, it's not temperature anymore, we actually have flow speeds. So I swam these fish at two different rates. I think it was 1/3 and 2/3 of their overall mean U-crit. So this is 13 centimeters per second and 26 centimeters per second. On the y-axis, we have tail beat frequency in hertz. So I actually filmed them with high frame rate and slowed down their tail beats so that I could measure how many tail beats they had any given second. Again, what we see is that the cold acclimated individuals are capable of, sorry, just hold on a second. Sorry about that. So what we see is that the cold acclimated individuals here are capable of higher tail beat frequency than the warm acclimated fish. And again, when we make them hypothyroid, we see the same trend repeated, where the cold acclimated fish have significantly reduced tail beat frequencies compared to the warm acclimated fish. So looking at the level of contractile function, here we are looking at SERCA activity on the y-axis. So SERCA is an enzyme that's involved in calcium cycling. And calcium cycling is really important for muscle cells because it regulates excitation contraction coupling. So in other words, SERCA modulates resequestering calcium back into the sarcoplasmic reticulum. So you have the sarcoplasmic reticulum, it releases calcium to stimulate muscle contraction, And then Circ is the enzyme that uses ATP to take that calcium and replenish the stores in the sarcoplasmic reticulum. So it's important to promote relaxation, but it's also really important, again, to replenish that calcium store for subsequent contractions. And that means that it's really important in fatigue resistance. So in cold environments, we see that cold acclimated fish have actually increased circa activities compared to cold acclimated hypothyroid fish. So thyroid hormone is acting at the level of this regulator of calcium cycling, presumably to improve rates of excitation contraction coupling, and possibly also fatigue resistance in cold environments. And again, I haven't shown this for the past few examples, but in all of these experiments, I also looked at the effect of supplementation with thyroid hormones in the cold acclimated hypothyroid fish. And again, we can see that if we supplement these hypothyroid fish by putting thyroid hormones in their water, we see almost a complete recovery of circa activity. So again, this was true in the tail beat frequency and the aerobic scope stuff that I showed you earlier. So this is a figure that I just made for an invited review from molecular and cellular endocrinology. And essentially what I've done here is kind of like broken down what I found in my PhD. So all the effects of thyroid hormone on these peripheral levels of performance, specifically in response to cold acclimation. So I didn't show you the data for all of this, but I'm going to go through it really quickly. I found, of course, that thyroid hormone, specifically during cold acclimation, promotes calcium cycling, both in cardiac muscle, also in skeletal muscle, presumably to increase muscle function in cold environments. I also found that it acts presumably on the preoptic area of the hypothalamus to increase sympathetic output to the heart. And that's going to support higher heart rates during maximum activity. I found that it also increases specific metabolic pathways in skeletal muscle tissue. So I'm showing the LDH pathway here. And of course, as I showed you, it also increases rates of transcription, so presumably rates of expression of these important metabolic genes. And it seems to be doing this as a regulator specifically during the cold to support higher aerobic scope and increase swimming performance. So what was interesting here is that even though I showed that thyroid axis is a main regulator of cold acclimation in fish, when I actually measured thyroid hormones in these fish, it was pretty unexpected. I would have expected the cold acclimated fish to have higher levels of plasma thyroid hormones than the warm acclimated fish, but what we saw was the reverse. And that got me interested in specifically how thyroid hormone is regulated in these peripheral tissues. So I don't have a ton of time now to go over this, but in 2016, I wrote this kind of nice overview of how, because thyroid hormone has so many tissue-specific functions, central like circulating levels, plasma levels of thyroid hormone aren't necessarily kind of the most important thing. So specific tissues have at least, or specific tissues and cells, I should say, have at least 4 or 5 different levels of regulation that can change how they respond to circulating levels of thyroid hormone. In response to this work, I also came up with this compelling idea that, idea that I thought was compelling, where thyroid hormone mediated cold acclimation in ectotherms. And this is an intermediary step in the evolution of endothermy. So this is really interesting because it looks like thyroid hormone co-opted the same levels of physiology in ectotherms that later were used to promote metabolic heat production or thermogenesis in birds and mammals. And this was specifically in response to the cold. So we've been able to identify at least one of these levers so far, but what can these levers tell us about costs of plasticity? So this is where I'm going to shift over to the next part of the talk. So how can we use an understanding of these molecular levers to start making informed predictions about the costs of plasticity? So for example, from the work that I just showed you, we know that the thyroid axis is an important regulator of cold acclimation in fish. But there's also evidence that thyroid hormone, or this lever as I should call it, is heavily used. So for instance, we know that thyroid hormone also regulates plastic responses to things like salinity, hypoxia, pH, and nutrition in fish. So if plastic responses to other environmental stressors also act through something like the thyroid axis or have overlaps in downstream pathways, then what happens when environmental conditions occur in a combination that starts pulling these levers in different directions? So I did a study to model this using bisphenol A, or BPA as it's more commonly known. So BPA is a widespread plastic pollutant that's essentially ubiquitous in the environment. So it was used in almost all manufacturing processes. Now they've replaced it with derivatives that are arguably worse, but it's really ended up in a lot of our aquatic environments. It's estimated even that over 95% of the world population has detectable levels of BPA in their bodies. So that's how common this stuff is. So this is kind of a proof of principle study that I used. And I specifically focused on endocrine disrupting pollutants, so BPA, because they represent a really good starting point to start looking at these stressor interactions. And that's because we already know the physiological pathways that they act through. So BPA has been long known to be an estrogen agonist, meaning it activates estrogen receptors. But in the last decade or so, it's also been shown to disrupt thyroid receptors. So this presents an interesting kind of like bottom-up angle that we can take to start predicting stressor interactions. Because we know the thyroid axis plays an important role, again, in cold acclimation, then it should follow that the toxicity of BPA is temperature specific, so that it's more toxic, presumably in cold acclimated fish. So this is what I predicted for the last chapter of my PhD thesis. And just for time's sake, I'm just going to show you a couple figures here, now focusing on cardiovascular physiology. So I did find some evidence that BPA is more toxic to fish who are cold exposed. For instance, it decreases maximum heart rates. So you can see that here in fish tested in this colder environment, that heart rate is decreased. I know this doesn't look like a huge decrease, but it's actually in the range of a 10% decrease in maximum heart rate. This is especially important at kind of the maximal end of performance, because we are already dealing really close to kind of the threshold of limitations, where a 10% drop in heart rate will have even bigger downstream effects on things like heart rate, scope, oxygen supply, and swim speed. So this isn't necessarily a linear effect in terms of its role with downstream functions. So looking again at calcium cycle, oh, sorry, I should mention, of course, that we see relatively no effect of BPA on maximum heart function in warm acclimated fish. Looking again at calcium cycling, we see this exact same trend. So again, we have circuit activity on the y-axis. That's the circuit is the one that resequesters calcium into the sarcoplasmic reticulum. What we see is a decrease in cold acclimated hypothyroid fish, again with relatively no effect, even maybe, I mean, this isn't significant, but possibly an increase there in the warm acclimated fish. So this is showing that we are able to use kind of this more like mechanistic knowledge of signaling pathways to really start identifying stressor interactions. Okay, I think I'm going a bit slow here, so I'll try and like speed it up a little bit more here, and then you guys can ask questions at the end if you feel like I've kind of not done something enough justice. So essentially, there's two ways that BPA can act to undermine the plastic response. It can just jam up the molecular machinery, in which case we don't, even though we get a cold acclimated fish, we don't get a cold acclimated phenotype. So this is just like jamming up the molecular machinery so that we don't get any kind of phenotype at all. You wouldn't necessarily expect any costs associated with this individual fish unless there were costs associated with the capacity for plasticity more generally. Alternatively, we could have a successful cold acclimation response, but these cold acclimated individuals, like for instance, this is all theoretical. So say cold acclimated individuals were characterized by increased thyroid receptor densities, then these cold acclimated fish may be more vulnerable to the effects of BPA. So throughout my PhD, I wasn't able to test whether or not BPA treated cold acclimated fish would display more of a maintenance cost if they exist or an ecological cost. So I'm going to shift into work that I'm kind of getting off the ground right now, and that's using these mechanistic understandings to predict ecological costs. But in order to do this, again, we really need to know which levers are pulled for which environmental stressors. And I've chosen to focus specifically on nuclear receptors. So nuclear receptors are receptors that function obviously as transcriptional regulators, but they also respond to small signaling molecules, so endogenous or exogenous signaling molecules. And they thereby represent ideal candidates to integrate environmental inputs with cell and tissue level responses. It's also interesting because many of them are already known to be involved in a number of plastic responses to environmental stress. Thyroid receptor is a perfect example based on what we just talked about. So nuclear receptors generally act to either repress or activate gene transcription. So they bind to a response element in the promoter of target genes, whereby they recruit cofactors, so co-activators or co-repressors, in the presence of their specific ligand. And that's going to, in this case, promote expression of the target gene and the target protein. So essentially what I'm doing here is building nuclear receptor activity reporters. So this is kind of parallel to traditional reporters, only instead of reporting expression levels, this is reporting functional levels of activity. So what I've done here is cloned the nuclear receptor response element into a minimal promoter. So this means that the promoter is not constitutively active. It's only turned on when it's activated by these transcriptional co-activators. And this is hooked up to the coding sequence for green fluorescent protein. What this really allows us to do is to look at specific nuclear receptor activity levels, where the nuclear receptor binds to the minimal promoter. In the presence of a ligand or co-activator, it will induce transcription or activate transcription. And what we see is expression of green fluorescent protein. So this is an estrogen receptor activity reporter fish that I've previously made. So we can see in control conditions on the left here, we don't get much GFP expression at all. And when we treat with an agonist, we see an increase in GFP activity, and that reflects the intensity and anatomical distribution of estrogen receptor activity. So the idea here is that I'm using this technique where instead of, previously I used this during a postdoc at University of Toronto to screen for small molecule libraries for pharmaceutical companies, but instead I'll be using it to screen for environmental stressors. So again, this is in progress right now, but what we might expect is if we looked at thyroid receptor, that during development, we might see activation of thyroid receptor in tissues like heart tissue, whereas we wouldn't see that effect in warm. Or in terms of reversible acclimation, we might see thyroid receptor activation, again, in cardiac or skeletal muscle, more during cold exposure than warm exposure. So the idea here is to create different nuclear receptor activity reporters and use them to isolate or identify which pathways environmental stressors act on. So we really can use a mechanistic understanding to predict what happens when these levers are overused or when they're pulled in different directions. But what about when these levers are underused? So what happens in terms of costs when these levers start to rust? So at this scale, we really need to take a different approach. We have to drop that treatment level effect that we looked at earlier and start looking at the level of the individual, specifically individuals that are flexible versus individuals that are relatively inflexible. So I'm going to talk about a bit of work which is also ongoing. The data that I'm showing you is preliminary, so I have a sample size of 8. I'm just going to say that right at the start. And I'm currently updating or increasing that sample size, but also kind of enhancing the platform that I'm using to measure these things. But essentially, what I've decided is to shift gears here and focus on starlet anemones. And I've done this for a few reasons. So starlet anemones are found, they are these tiny little burrowing anemones that burrow into substrates at the bottom of saltwater marshes. And then they raise their tentacles above to feed on floating particles in the lower parts of the water column. So essentially, they are found natively all up the east coast of North America. They've been introduced in certain regions on the west coast. And because of this huge latitudinal gradient, they're found at an extremely large range of temperatures. They've been found between 1.5 and 32.5 degrees Celsius. They also experience huge seasonal and diurnal variations in temperature, as you can imagine, again, with this huge latitudinal gradient. I specifically chose to focus on them because they reproduce sexually and asexually. So we can actually promote asexual reproduction simply by cutting them in half with a razor blade. And over the course of a week or two, depending on the temperature that they're regenerating at, they'll actually regenerate their entire body plan. So you end up with 2 clones of a single genotype. So we can get entire colonies of anemones that all originated from a single, that are all clones of a single genotype. So in terms of this preliminary work that I'm going to show you, I looked specifically at routine metabolic rate in these guys. And this is because during development, they are rapidly growing marine invertebrate, where metabolic rate is predicted to scale with growth rate, and growth rate has obvious fitness implications. So we're currently testing kind of the effects of growth rate, the links between metabolic rate, growth rate, and fitness in these guys. I don't have that data for today's talk. But essentially for our experimental design, what I did is clone individual genotypes of these guys. So I cut off the very aboral end here with a razor blade. And I did this and developed clones at 3 different developmental temperatures. So 16, 21, and 26 degrees Celsius. I then grew them under common garden conditions. So at this 21 degrees Celsius temperature, at which case I reversibly acclimated each clone to 3 different temperatures reflecting the developmental temperature range, and then tested thermal performance curves, again for routine metabolic rate across a range. This is 3 temperatures here. I actually did it across a range of 4 temperatures up to 32 degrees Celsius. Up to 31 degrees Celsius. And again, I have a sample size of 8. So in terms of raw data, what I'm left with is all these thermal performance curves. So each color denotes a different genotype. So we have the different clones horizontally, and then the different reversible acclimation treatment for each clone vertically, where shifts in thermal performance curves between these columns and rows indicate either developmental plasticity, or reversible acclimation. So this is just to give you an overview of all the data that we can collect. And this is, again, only with a sample size of 8. I can then use those shifts in thermal performance curves to start identifying the capacity for plasticity of individual genotypes. So in terms of developmental plasticity, we can take an individual where these curves shift a lot. I've been quantifying the area between these curves to indicate just how much these curves shift. So this one would have a high score of developmental plasticity, whereas individual 2 would have a relatively low score of developmental plasticity. And that's because its physiology doesn't seem to be very responsive to the thermal environment in terms of acclimation. Okay, for reversible acclimation, we can do the same thing. Again, where we have individual genotypes that show a high degree of thermal plasticity, again, through shifts in these thermal performance curves, whereas we have other individuals like individual 4 here that have a low capacity for plasticity. So by identifying these capacities for plasticity for given genotypes, we can start to ask questions about costs. One of the cool things I thought we could look at is whether or not flexible individuals can bend both ways. So are individuals that are specialized for cold acclimation also specialized for warm acclimation? Or does that represent a cost or a trade-off of plasticity? What we see with developmental plasticity is that those individuals that are most developmentally plastic in terms of cold exposure are also most developmentally plastic in terms of warm exposure. So this was just on the threshold of significance there. And again, sample size of 8. So kind of take that in the context of the experimental design to this point. In terms of reversible acclimation, we see with the sample size of 8, this really nice trend, arguably driven by this one data point over here. But I mean, there's evidence that individuals that are good at cold acclimating are equally good at warm acclimating. So it does appear that plastic individuals bend both ways in response to temperature in the system. Looking at performance costs, so what I've done here is looked, again, specifically at developmental plasticity. So on the x-axis, we have developmental plasticity score. The higher the score, the more developmentally plastic they are. And on the y-axis, we have performance score. So this is going to be routine metabolic rates. And this is from individuals, or this is from clones that developed and were acclimated to 21 degrees Celsius. So what's the cost of being a plastic individual when you're in an environment that's not necessarily requiring that plasticity to be used? And what we see is that more developmentally plastic individuals have lower performance. So their routine metabolic rates are lower. So again, we were testing the downstream effects of this on things like digestive efficiency, growth rates, traits to get us closer to how this will affect the fitness of these individuals. We don't see this trend in terms of reversible acclimation. So there doesn't seem to be this same trade-off between capacity for plasticity and performance. Looking at another possible trade-off, I've looked here at thermal sensitivity. So this is the Q10 effect. This is how much performance changes over a 10-degree drop or increase in temperature. So having really high thermal sensitivity is bad when you have shifting environments, because that means that even if you have optimal performance at the ideal temperature, as soon as things shift a little bit, you're going to see huge decreases in performance. So I found no evidence of a trade-off here with developmental plasticity, but what we see is this really nice trend, again, with reversible acclimation, where individuals that have higher capacity to acclimate also have higher thermal sensitivity, which isn't necessarily a good thing. Now, this also is kind of logical because you would expect the individuals with higher thermal sensitivity, so that's the individuals with like really narrow thermal performance curves, they're the ones that are going to need to have high plasticity to compensate for changes in temperature, because changes in temperature are going to affect those individuals the most. So it might not be entirely surprising to see this trend. So we found that plasticity does in fact come at a cost, particularly when that flexibility is not being used or when it isn't needed. So as I said before, I'm currently linking these routine metabolic rates to kind of more fitness related traits like growth rates, feeding rates, cost of digestion, so specific dynamic action, and also rates of asexual production. And ultimately, I'm actually working on my NSERC Discovery Grant proposal right now. But these guys are also a really emerging biomedical tool, and we can actually do transgenics in them. So the idea is to make the same kind of nuclear receptor activity reporters that I was talking about in zebrafish, in these guys where we can identify flexible and inflexible genotypes. It's also really cool because they represent obviously an evolutionarily early animal. So we only see a subset of the nuclear receptor repertoire. So we don't get all these like isoforms with various functions and stuff. So this really will give us a good idea of how I guess, how these nuclear receptors respond to environmental stress in a more simplified system, if you will. So I just want to finish up by bringing this back kind of to where I see my role in this like bigger framework in terms of collaborating with different people in labs. And again, that's where I focus on these gene environment interactions, specifically trying to identify these molecular levers so that we can understand maintenance and ecological costs. I think this integrative framework is a really powerful approach because as opposed to assaying kind of every combination of stressor interactions from the top down, we can start kind of building this more informed framework from the bottom up that will really help us to kind of identify overlaps in these pathways, and that should help us predict stressor interactions. So rather than this more like phenomenological approach of looking from the top down. Okay, so I just want to give everybody a big thank you that was involved in this work, also the funding bodies, and to everybody who showed up today to listen to my research. And I mentioned this like just in conversation with the organizers here at the start, but I'm also looking for prospective grad students next fall. So if anybody finds this work particularly interesting, feel free to reach out and we can discuss possibilities. Also postdocs too, obviously. I don't specifically have postdoc funding, but I'm more than happy to help craft applications for funding. So with that, I just want to thank everybody again.
Cool. Thanks, Alex. It was a great talk.
Yeah, of course. Thank you.
So how we're going to do questions, if people have a question, if they just want to like throw I have a question or I have a comment in the chat box, and then we just go in order from there.
Sure.
So
I'll go back to sharing my screen, I guess.
cool. Well, I guess I'll start. I have a question for you.
Sure.
Do you think there's potential for long-term costs associated with cold acclimation for something in like zebrafish? So like, will their aerobic scope decrease after like X amount of time, and could there be like overcompensation for that?
Yeah.
Thyroid-mediated acclimation?
I think that there probably are. I would predict that, especially if environments got colder, there would be costs associated with that. But I think, and there's not like a ton of empirical evidence to back this up, but I think that these animals have likely evolved kind of thermal thresholds over which temperature, and like, I guess if we were talking about cold, over which temperature acclimation responses make sense and are beneficial, and kind of like you get the bang for your buck, but under which I think that they probably shift more from an acclimation response to like a passive response, like something like dormancy, where they start shutting down their metabolism. So I think there probably are costs associated with long-term cold acclimation, or like if I had gone 5 degrees or 10 degrees colder, we wouldn't necessarily see the same thing. But I think those costs are such that like populations that have historically or evolutionarily experienced those things have probably compensated for by employing a different strategy. Another good example, though, that like for that is mismatch costs. So some people like pick this out when I show them the talk, but if we go back to just the swimming acclimation here, you can see that the cold acclimated fish swimming in the warm test temperature actually outperform the warm acclimated fish. This is specifically why I don't like, I caught myself here, but I said like cold acclimated fish have better swimming performance. Really, I don't like saying that. They have faster swimming performance. But whether or not that's beneficial at this warmer test temperature really isn't known. I haven't actually, because over time they like reversibly acclimate back to that warm acclimation phenotype. But I think that really suggests that there are costs, like mismatch costs associated with these higher rates of swimming, and presumably all the physiological mechanisms that have been adjusted, where either there's not selective pressure in warm environments to attain these levels of swimming, or where you just have kind of this imbalance in like metabolic pathways where it's just not necessarily efficient. So yeah, I think there's definitely costs associated, whether they're mismatch costs or more metabolic costs.
Yeah, that would be interesting. I'm just thinking about like thyroid hormone action. Like maybe if there's that compensation, maybe there could be like desensitization of to like receptors and cells, or
Yeah,
yeah,
I wonder that. And I mean, it's like, yeah, that's actually the perfect question to use those thyroid receptor activity reporters with too. Like we can see over time in these cold acclimated fish, whether or not those receptors are still lit up like you imagine they would be early on in the acclimation process.
yeah.
Yeah, that's a really good question.
It's like Adam, you had a question, but it got answered. So cool.
I could ask another one though, if you don't mind.
Yeah, go for
Yeah,
it.
of course.
So hey, really enjoyed the talk.
Thanks.
So I also did my MSc at Queen's, doing something a little bit similar with Daphnia, although I was more on the like phenotype to fitness side of your summary there.
Cool.
I'm curious, with these animals, how much do you think like where they are there's reverse acclimation and they can within one lifetime move back and forth between like optimal like metabolic rates and things like that? How much do you think that limits like their actual acclimation status limits like performance decisions or behaviors in nature? Like even if in the lab they can, if you force them to switch, they can switch. How much do you think like in nature when they, you know, they make it developmental, they go down one trajectory, how much do you think that limits like behavioral switching or I don't know, like feeding preferences and things like that?
Do you mean in terms of like developmental plasticity?
Developmental plasticity, or like say they acclimate to a cold temperature and maybe there's like behaviors that are associated with cold temperature, but they could also acclimate to warm temperature. Do you think like outside of the lab, do you think there are instances where one individual will like go back and forth between like cold tolerance or warm tolerance in nature?
Oh, yeah, I think so for sure. I expect so. I mean, I would guess that individuals with kind of longer generation time relative to like seasons will definitely kind of employ this like reversible acclimation response, where you'd see them seasonally like acclimating to different temperatures. So that's kind of been shown, I guess. It's hard to tease apart like how much of it is like specifically temperature driven as opposed to something like photoperiod and stuff like that. But I mean, in a general sense, what you do see is like reversible remodeling seasonally. Now, with something with like a shorter lifespan, like I worked a little bit with mosquito fish, which, yeah, I guess zebrafish, I actually don't know how long they live in the wild. In the lab, they'll live like 14 or 15 months, but don't look like particularly good by the end of it. But yeah, I think it depends on the environment mostly, whether or not. So in terms of like developmental responses in animals with relatively short generation times, I think you tend to see more like canalization of these traits and these behaviors where they kind of just put all their money on like kind of all their chips on like one number. Whereas, yeah, I think in animals with longer generation times, I think they certainly experience these like reversibly plastic responses in the wild.
Yeah, that was my sense with working with Daphnia too, because they're fairly short-lived, right? And so even
Yeah,
in the lab, you can have them up and down regulate hemoglobin, which like
yeah,
has to do with like oxygen and yeah, respiration. But in nature, yeah, that was my sense as well, that they kind of just pick a trajectory. Anyway,
yeah,
thanks very much.
cool.
Any other questions from the audience out there? You can always just chime in if you don't feel like typing in the chat box. I guess I'll ask another question because I have a lot.
Sounds good.
So really, I wasn't quite expecting the shift from fish to cnidarians, which is really
Yeah.
interesting. And you kind of mentioned a little bit about they're going to be lacking these subtypes and all these receptors because they're lower on the tree. But so like, I mean, do they have like similar like thyroid receptors? Are there like homologous receptors between fish?
There's not really a homologous receptor for thyroid receptor per se. So humans have, I think, 46 nuclear receptors. Zebrafish have 72 nuclear receptors, and these starlet anemones have 17 nuclear receptors, so like way fewer. So there's no thyroid receptor homolog. The closest thing you'd find is retinoid X receptor,
Okay.
which in in vertebrates, and especially higher vertebrates, thyroid receptors and like retinoid X receptor, RXR, kind of cooperate. They have, yeah. So in some sense, I wouldn't call RXR homolog in these guys, but it's probably based on phylogenies. It looks like one of the RXR copies through the whole genome duplications in animal evolution eventually turned into what we call thyroid receptor. So yeah, so that's one of the main receptors. I kind of like identified 3 receptors to start building these activity reporters for in Nematostella or the starlet anemone, and RXR is like definitely one that I focused on for those reasons. Cool. Yeah.
Any other questions from the audience?
People like, I mean, if you think of questions later too, like always feel free, like they can email them too.
Sure, yeah, that's a good idea.
Yeah, I'll put that last slide back up just so.
Cool. Well, I guess I have one more question, and if people are out of questions, then we can end it there. But um, I was, I mean, there's more and more popping up in the literature about thyroid hormone having all these non-genomic actions.
Yeah.
And so, how much of a role do you think? I mean, I guess you didn't measure it, but how much of a role could that have been playing in your um, in all like your different measured tissues in your cold acclimation experiment? Or I don't know if you
Yeah,
measured, if you just used T3 or if you're using
we
other
looked at,
thyroid.
yeah, I didn't get quite into that. So we looked at T3, but also T2, which is like a derivative of T3. So T3 is like the kind of classical active thyroid hormone. And then T2 is a derivative, which they've like recently been shown to have like physiological activity. So I looked at both T2, T3. I mean, in some cases, I saw a distinction between the two, but in lots of cases, they both kind of had the same effect. Um, in terms of the genomic action, or sorry, the non-genomic actions, I think that that was kind of the original hypothesis for T2. So in terms of the metabolic gene transcription, so the RNA expression levels for that, for instance, I kind of only expected a response with T3 because I thought that T2 would act through more like non-genomic means, where it would act either like directly on the mitochondria or like I think there's still like unresolved kind of extranuclear pathways for it. Surprisingly, what we did see is, and I mean, yeah, I should go back and look at that figure, but if I remember correctly, for most of those metabolic genes, there was kind of the same increases in transcription between T2 and T3,
Okay.
which is really interesting.
Yeah.
So whether or not T2 also acts, well, I mean, T2 must also act transcriptionally, whether or not that's direct or indirectly through kind of non-genomic actions, I'm not really sure. But I mean, again, that would be a really cool system for the activity reporters because you could apply T2 and see whether or not that like lights up the thyroid receptors and how quickly it lights them up.
Yeah.
Yeah, but yeah, somehow T2 is still regulating transcription, even though its actions have been described as like more strictly speaking, like non-genomic in nature.
Yeah, that's because that's how it sounds in the literature also. So
yeah.
Cool. Well, if people have no more questions, I think we could just end it there.
Cool. Well, thanks everybody.
Yeah, that was awesome, man. I'm really glad that you came along. Or
thanks, Ali.
You got to come.
Oh, thanks for having me.
Maybe in the future we'll fly you over here for another seminar.
Yeah, once I do like some more of this work, that would be awesome.
Yeah. So cool.
So do you work at all with thyroid hormone, Cole?
Yeah. So me and my supervisor are looking at interactions between appetite and thyroid hormones.
Oh, okay. I have seen that research. I just didn't put it like together with you.
Yeah. So
okay,
yeah,
it's
kind of cool. So
yeah.
But yeah, it's interesting because you're talking about in your talk how like peripheral circulating levels of thyroid hormones it's not a good like tell all.
Yeah, I think so. I mean, I kind of wrote the review that I talked about in 2016 was kind of like a review that I wished I had when I did my PhD. That was kind of like when I was starting my PhD, that was kind of the theme behind it. Essentially, I went into it just like knowing thyroid hormone kind of from like undergrad classes, and that it's like regulated through like negative feedback through the like central nervous system, like pituitary axis. But yeah, I think it's like when you think about all the crazy different like tissue specific responses you see, it must be mainly regulated at a peripheral level. And kind of what I would expect is that as long as circulating levels of thyroid hormone aren't limiting, then I would expect that it's peripheral tissues that kind of decide what effects it actually has.
Yeah, absolutely.
Yeah, the review paper that I just submitted that like review or commentary to like molecular and cellular endocrinology. And that I did more of like kind of a wide scale survey of people's thyroid hormone studies in like different ectotherms. And it does look like, in terms of people's experiments, it looks like making hypothyroid animal or making animals hypothyroid has a lot more of an impact than making them hyperthyroid. And
Mm-hmm.
again, I would expect that that's because peripheral tissues are the ones that decide like how much thyroid hormone they like transport into the cell, and then how that's kind of metabolized and used.
Yeah, totally.
Yeah.
Yeah, yeah. I just think thyroid hormone is it's just such a powerhouse hormone. You know, it's so cool.
Yeah, it's crazy. Have you come across, there's a woman uh, who's a little bit like eccentric. Well, I mean, I gather that from what she writes. I've never like met her personally. What's her name? Crockford, I think, she's in Victoria, and
Okay.
she wrote like this giant paper, essentially, I think it was in like maybe like 10 or 15 years ago, saying that thyroid hormone is essentially like the single agent that's responsible for like speciation, which,
Whoa.
yeah, is a pretty big statement to make. And then, but I've always thought she had like really cool ideas. So I like reading her stuff. But since then, she's like drifted into like really unusual territory in terms of like denying climate change and stuff like that. So
Interesting.
anyway, yeah, but I mean, it would be worth, I'll send you the paper, because there's like, as crazy as the big picture is, she's like, you can tell that she's like super on it, super intelligent, and like has lots of like crazy interesting ideas in there.
Interesting. Okay.
But big picture, it's kind of bullshit.
Yeah. Oh man. Huh.
Yeah, I actually don't know. I think they may have gotten rid of her, the like University of Victoria.
Oh really?
Yeah,
Wow.
I think possibly because of the climate change stuff, which